Good morning, everybody. Welcome to this week's installation of Microseminar. It's my pleasure to introduce Dr. Kanika Khanna, who is at Stanford University, just starting a new postdoc there. She originally, sorry, I had the, there's the, sorry. I had the bio in front of me and then I lost it. So Dr. Khanna is originally from New Delhi and did a bachelor's and a master's degree in biochemical engineering and biotechnology from the Indian Institute of Technology in Delhi. She then went to do a PhD in biology at UC San Diego in the laboratories of Dr. Elizabeth Villa, Kit Pogliano, and Joe Pogliano and finished up in January 2020. And so during that time, she did a lot of research on Bacillus subtilis sporulation, phage replication, and she's now transitioning to more of an ecology type of research at Stanford with Dr. Michael Fischbach to understand mechanistic or to explore mechanistic understanding of host microbiome or interactions in the human gut using model systems in mice and stable synthetic communities. And so some of that research coming up is going to be really exciting. So I have jackhammering going on, so I'm going to make sure I turn it over to Dr. Khanna and let her regale you so you don't have to listen to any more of me. Dr. Khanna, take it away.
Yeah. Hello, everyone. Thank you so much for the introduction, and I'm really excited to talk to all of you about my work, which I did during my graduate school at UCSD in the labs of Professor Kit Pogliano and Professor Elizabeth Villa. And I'm just going to start sharing my screen. Right. Yeah, so the work that we did was studying the cell division machinery or the mechanism of cell division in Bacillus subtilis during sporulation and during normal vegetative growth. And the work that I'm going to talk about actually just got published or as a preprint in bioRxiv last week itself. So I would encourage all of you who are interested in reading more about the work to look at this preprint, which we have submitted. And so coming back to bacterial cell division, now all of us understand cell division in bacteria as a very simple process of binary fission, where the division site is placed at the middle of the cell and the cell divides into two equal parts or into two equal daughter cells. But the mechanistic details, or what are the proteins which are involved in the cell division machinery, or the proteins which constitute the divisome of B. subtilis, we don't understand a lot about what these proteins are, how their interactions with each other and with the other components of the cell are. So what we know so far is that the major protein which is involved in cell division in many bacteria and also in B. subtilis is FtsZ. So FtsZ is a homolog of tubulin, and it can polymerize in a GTP-dependent manner. So when the division site is placed at the middle of the cell, what happens is that FtsZ forms a ring, which is made up of overlapping protofilaments of FtsZ, and this ring is called the Z ring. And the Z ring acts as a scaffold to recruit other cell division proteins to the septum. And this happens in two stages in Bacillus subtilis. So in the first stage, proteins which promote the interaction of the Z ring with the membrane are recruited. And in the second stage, proteins which are synthesizing the cell wall in the septa or the peptidoglycan in the septum are recruited to the Z ring, or directly or via interaction with the other cell division components which are promoting the interaction of the Z ring with the membrane. And you don't have to remember all the proteins for this purpose of this talk, but I'll be talking a little bit more about FtsA. And what is special about FtsA is that it is an actin homolog, and it also has the ability to form filaments both in vitro and in vivo. So this is a time-lapse movie, which I'm going to show, and you can see here FtsZ, which has been fluorescently tagged. And if you look at any one of the cells as the time-lapse goes through, you will see how the process is initiated in space and time. And as you can see here, if you concentrate on this cell, what happens is that the Z ring first constricts. And as the Z ring constricts, the cell divides into two, and then new Z rings are placed at the new daughter cells, which are going to divide further. So basically, cell division is a very, it's a process which involves coordination between the constriction of the Z ring, the synthesis of the septal cell wall, and the division of the cell into two equal daughter cells. Now, This is really nice. So, that Bacillus subtilis can divide into two daughter cells. But what is special about this bacteria and why we are interested in studying in this, Bacillus subtilis is in our lab is that Bacillus subtilis has another pathway which is called sporulation. And this pathway is initiated when the conditions in the environment are deprived of nutrients, so under stress conditions. And what is special about this pathway is that instead of having the division site in the middle of the cell, the division site is placed close to a pole. And this leads to an asymmetric division event because the two cells which are formed are different in sizes, and ultimately they also have different fates. So the smaller cell is called the forespore, and the larger cell is called the mother cell. And eventually, what happens is that the mother cell membrane moves forward in order to engulf the forespore. And once the spore is completely inside the mother cell, it is protected by different layers of coat and cortex, and which also preserve the genetic material of the spore inside this endospore. And eventually the mother cell will lyse and the spore is released, and these spores can stay dormant for many, many years. I mean, there are reports of isolation of spores which are as old as 25 million years ago. So it's a and once the conditions in the environment are favorable again, there are nutrients and everything, these spores can germinate back to divide vegetatively. So sporulation is a very relevant process because many of the spore formers, they can cause diseases because they can hide their cellular machinery, so to say, when the conditions are not favorable. And when the conditions become favorable, they can release the toxins in their respective hosts. So B. anthracis, which is known to cause anthrax, Clostridium botulinum, or B. cereus, which are known to cause food poisoning. And also about 30% of the microbes which are present in our gut are spore formers. And we still don't know a lot about the physiology or what these spore formers are doing in our body. So it's a very exciting area of research in general. So coming back to cell division, so we know that Bacillus subtilis can divide in two ways. So during vegetative growth, when the division site is placed at the middle of the cell, and during sporulation, when the division site is placed close to a pole. Now, apart from the fact that these division sites are placed differently, another difference is that the thickness of the septal cell wall is also different in both the cases. So the vegetative septum is around 80 nanometers thick, and the sporulating septum is 1/4 the thickness, which is around 20 nanometers thick. And recently, there have been studies which have shown that the cell division protein, especially FtsA, is involved in how much septal cell wall is going to be incorporated during division. And one of the mechanisms that has been proposed is that these FtsA filaments, as you can see in the schematic here, in red, they are treadmilling, which means that new subunits are being added at one end and old subunits are removed from the other end. And this leads to a constant treadmilling motion of FtsA around the entire circumference of the cell. And what this motion of FtsA does is that it moves the enzymes which are making the septal cell wall, which are shown here in blue, around the circumference of the cell to make a new cell wall. So there is a link between the cell division machinery and probably how much or what is the amount of septal cell wall being incorporated in the septum, but we don't understand clearly what these relationships might be. So these are some of the questions that I wanted to ask when I set out working on this project. The first was, can we look at the molecular organization of these FtsA filaments during vegetative growth and during sporulation and see if there are any differences, because these are essentially two different ways in which the cell is dividing. And then if we do see any differences, can these differences explain why the septal cell wall is different during vegetative growth and during sporulation? And if that is the case, then can we understand what are the factors which might regulate the divisome architecture during vegetative growth and during sporulation in Bacillus subtilis? Now, in order to address these questions, we wanted to go to high resolution microscopy methods. And one of the reasons is that these bacteria are, if you get a little bit more nerdy into the numbers and everything, so these bacteria are like 1 micron in size or so. And the current optical microscopy methods, namely fluorescence microscopy, they only offer a resolution of around 200 nanometers or so. So we're really not able to understand these processes at a very high resolution in bacteria. As you can see here, this is one cytoplasmic protein which has been GFP tagged. And you cannot make any inferences about the localization of these proteins using optical microscopy alone. And recent advances have placed super resolution microscopy, where you can technically resolve up to 20 nanometers or so. But again, you won't get much information. And we are especially interested in looking at the septum, which is like 80 nanometers or 20 nanometers. So we really need high resolution microscopy methods if you want to see individual filaments or individual monomers of proteins. So I'm going to talk a little bit about this technique, which I've been working on, called cryo-electron tomography. And it really allows us to zoom inside the cell and see these proteins, individual protein complexes, which are a few nanometers in sizes. And we can understand what are the relationship of these proteins with other proteins in its surrounding, and so on and so forth. So talking a little bit about this technique called cryo-electron tomography, which is a technique to visualize macromolecular complexes in situ. So I'm going to show you a movie in order to understand this process better. But as the name suggests, you are basically shining your bacteria with electron beams, and it is in cryo mode. So you are freezing your bacterial samples specifically. And the process of tomography, I'm going to talk through this video, but it's very much like CAT scan. So in this video, what you see are these virus molecules. So what we first do is that, we freeze them in a monolayer of ice. And once we have these, molecules in the layer of ice, we take them inside a transmission electron microscope. So what happens inside the transmission electron microscope is that you're shining an electron beam through your specimen, and you're rotating your specimen and taking projection images at different angles. So this is just like a CAT scan or an X-ray scan, if you've ever gotten one, where you have, you want to take three-dimensional view of your hand, let's say, or your bones and different structures. And once you have these all these images and different projection images at different angles, you can combine all of these images together in order to get an idea about the three-dimensional identity of the specimen. So just like you would get in an X-ray scan, but the only difference is that there your hands are continuously moving, versus in tomography, what is happening is the sample, in X-ray, the X-ray beam was continuously moving, versus in tomography, you have your sample of interest being constantly projected at different angles. And once you have these 3D images, you can see here that you can start annotating the different parts of the cellular specimen. So for example, in these viruses, you can start annotating the capsid or the glycoproteins and everything, and so on and so forth. So but one of the limitations of this technique of cryo-electron tomography is that it's not suitable for thick specimens, and Bacillus, which are like 1 micron or so, are also very thick for cryo-electron tomography. And in order to circumvent this, we incorporate another technique where we're actually shaving our specimens to make them thin. And this technique is called cryo-focused-ion beam milling. So this is a mammalian cell. And this is the inside of a focused ion beam milling instrument. And what, it's a dual beam. So there are 2 beams. One is a scanning electron microscopy. So all the pictures of the bed bugs and everything we see, we can take them using an SEM. And then at a certain angle, there's another beam, which is the focused ion beam milling beam. And using this beam, which are basically made of gallium ions, which are highly positively charged, we can actually, it's like bombarding your specimen continuously with high charges, and this will ablate the cellular specimen. So I again have a little video in order to really appreciate this concept. So again, you have this a grid where you have your mammalian cell, and what happens inside a cryo-focused ion beam or a cryo-fib, as I'll be referring to in most of my talk later on, the cryo-fib instrument is that, you can have this fib beam, which comes in patterns. So basically we have 2 beams and we have a 1 beam, but they're coming in 2 patterns. And using these patterns, we can ablate the cellular material from the top and the bottom of the specimen. And this leaves us with a thin slice of our biological specimen of interest. And it is supported on the sides by the cell itself. So it's a really cool technique to start imaging your bacteria at high resolution. So just to summarize what we do in our workflow so far is that we have these bacteria, which we freeze in a monolayer of vitreous ice. And by vitreous ice, I basically mean that we freeze them so quickly that water doesn't have time to form crystals. And it's like an amorphous layer all over the bacterial cells. And then we make them thin inside a cryofib instrument by using a beam of gallium ions. And once we have this fib milled specimen, we take it inside a transmission electron microscope, we shine electron beam through it at different angles, we take different projection images and combine to get a 3-dimensional view of the specimen. And this really allows us to view the molecular complexes at a resolution of a few nanometers in their native context. And this is an example of a tomogram of a sporulating Bacillus subtilis cell. And what you see in this movie are basically you're going through up and down through the different slices in 3D, which you have taken for your particular image. And then you can again annotate these different slices. So here you can see I've annotated the spore, the membranes, the cell wall. You can also pick out ribosomes, where they're located inside the cells, and some of the coat filaments and coat proteins. So coming back to the problem of cell division. So previously, this FtsZ ring, which is involved in cell division in bacteria, has been visualized using cryo-electron tomography in E. coli and in Colobacter crescentus. And so this gets a little bit really fun from here, because if you look at this dividing E. coli cell from Jan Loewy's lab, so what they see is that near the dividing septum, you see these dots. And I know it's really difficult. It took me like my whole PhD in order to visualize these dots. But what they saw was that these dots were not just any dots, but they corresponded to projections of filaments. So what I mean by that is that if you were to cut a plane which goes through the short axis of the cell, and then you rotate the cell by 90 degrees towards yourself, you will see a septal disc. And each of the dots which they see in this top view corresponds to a filament in the projected view. So here you can see, very bleakly this line which corresponds to an FtsZ ring in E. coli. And now we want to visualize this FtsZ ring in Bacillus subtilis. But one of the things which I want you to keep in mind is that we have this rod-shaped Bacillus subtilis cell. And if we cut a plane through the short axis and rotate the cell by 90 degrees toward our cell, we see a septal disc. But we are also fib milling our specimens. So what you will see are only parts of the septal disc. And again, we don't have a lot of control over where we are milling our cells. So depending on the angle of, how the cells are oriented on the electron microscopy grid, or how the beam is oriented with respect to the cells, we can capture different parts of the septal disc. So coming back now, we wanted to see, can we see these cell division proteins, these FtsZ proteins in Bacillus subtilis? And so we started taking images of dividing vegetatively growing Bacillus subtilis cells. And if you look, now what I'm going to show you is that I'm going to show you two views. So this is the zoomed in view of the left side of the invaginating septum. And this is a zoomed in view of the right side of the invaginating septum. And so I'm going to show you this part, which is going to be unannotated. And I'm going to annotate some cellular parts on the right side in this and all the subsequent slides. So just so, I can give you a comparison, what I see is also what you see in these images now. But if you look very closely, so in this peach color is the cell membrane, which has been outlined here. And near this invaginating membrane, we see not one, but two series of dots. So one is closer to the membrane here in pink, which we call the membrane-proximal series of dots. And another one is a little bit away from the membrane in blue, which we call the membrane-distal series of dots. And again, how do we know that these are not just any dots near the invaginating membrane, but they correspond to cell division proteins? We adopt the similar thing, that technique. We cut a plane which goes through the short axis of the cell, and then we rotate the cell by 90 degrees towards ourselves. And what do we see is that, so this is again the zoomed in view of the left side of the invaginating septum and the right side of the invaginating septum, but this time you're seeing the septal disc and not the top view. And again, in peach is the annotated cell membrane. And what you see here is in pink, the dots which we were seeing closer to the membrane correspond to these filaments in pink, which we are now the membrane-proximal ring. And the dots which are a little bit away from the membrane now correspond to this ring in blue, which is the membrane-distal ring. So this was really exciting that, we know that the proteins which we are seeing near the invaginating septum, they're not just any proteins, but they correspond to cytoskeletal rings, which are likely involved in cell division. And these are just the movies which are going through different slices of the tomogram. So you can see here, this is the cell wall, and these are the dots which are going, going in and out of the view as you're going through different slices. And here you can see in the projected view, these different, the filaments which are encircling the cell. And this is the cytoplasm, and this is the cell wall. So next we wanted to know what are these cytoskeletal filaments, because in B. subtilis, what we see is a membrane-proximal ring at a distance of 6.5 nanometer and a membrane-distal ring at a distance of 14 nanometer. Versus in E. coli, previously they've only seen one ring at a distance of around 50 nanometer, which corresponds to FtsZ. But another interesting thing is that when they overexpress FtsA, which is also a protein involved in cell division, and it's an actin homolog, and it anchors the Z ring to the membrane. So when FtsA is overexpressed in E. coli, they start seeing FtsA between the FtsZ ring and the membrane in E. coli as well. So this was our hypothesis that probably in B. subtilis, the membrane proximal ring that we see is corresponds to FtsA, and the membrane distal ring that we see corresponds to FtsZ. And in order to test this hypothesis, what we did was that we made a strain in which we modified the length of the linker region which connects FtsA and FtsZ. So we incorporated a glutamine-rich linker, and this will increase the distance of the Z ring from the membrane. And in this strain, then we imaged what is the distance of the membrane proximal ring from the membrane and the membrane distal ring. And what we saw was that the distance of only the membrane distal ring from the membrane increases, suggesting that the membrane distal ring is the one which corresponds to FtsZ, versus the membrane proximal ring, which corresponds to FtsA, because we don't know of any other filaments which form any other protein which form filaments in vivo and in vitro. And based on the results in E. coli, we assigned the identity of the membrane proximal ring to be that of FtsA. So just to summarize, the cytoskeletal filaments that we see in dividing vegetative Bacillus subtilis cell during vegetative growth are FtsZ, which is the membrane distal ring that you see here in blue, and FtsA, which is the membrane proximal ring, which you see here in pink color. All right. So this was really good, and this is just a short movie showing that you can annotate, these filaments inside the cell. So this is the cell membrane, and then you can zoom in and see these FtsA and FtsZ bundle inside the cell, which are going around the circumference of the cell, but you only see a section because we are essentially milling our cells. So this was really exciting that we can see how these cells are organized during vegetative growth. And the next question we wanted to ask is that, can we image these dividing cells and see the organization of FtsA and FtsZ filaments during sporulation as well? So we image dividing sporulating cells, and this is again a zoomed in view of the left side and the right side of the invaginating septum. And to our surprise, what we saw was that these FtsA and FtsZ filaments, they were only present on the mother cell side of the invaginating septum. So the smaller side is the forespore and the larger side is the mother cell, and these proteins were exclusively present on the mother cell side of the invaginating septum. So this was really nice because and just to confirm that these dots which we are seeing are not just any dots, but they correspond to cytoskeletal filaments. So if we take basically the cross-sectional view of these cells, we see that, this is the forespore and this is the mother cell, and these filaments which correspond to these dots are only present exclusively on the mother cell side. So this was really exciting that there is actually a difference in how the cell division machinery is localized during vegetative growth and during sporulation in Bacillus subtilis. So it's not just that the division septum is placed at different places during these two modes of growth, but also the cell division machinery is localized differently around the invaginating septum during vegetative growth and during sporulation. So the next question we wanted to ask is that, now we know that there exists a difference in how these FtsZ filaments are organized. Can these differences also explain why the septal cell wall is different during vegetative growth and during sporulation? So as I told before, that the sporulating septum, once it is completely formed, is almost 4 times thinner than the vegetative septum once it's completely formed. And there are models which propose that there's a link between the amount of condensation of the Z ring and the dynamic activity of the Z ring around the circumference of the cell, which dictates the motion of also the cell wall synthesis around the septum. So in order to investigate this, the first thing that we thought was, okay, maybe we can count the number of filaments during vegetative growth and sporulation. But that became difficult because even though cryo-electron tomography is a very high resolution technique, it's difficult to count the exact number of monomers because these proteins are placed just a couple of nanometers apart, and it's sometimes not possible to resolve all of them. So, but we know that, from our images that most of the times the distance between these two dots that we see is similar in vegetative growth and during sporulation. So what we did was that instead of counting the number of filaments, we measured the distance which is spanned by this bundle of FtsZ during the two modes of growth. And the first thing which we wanted to see was that we know that the septum is thinner in sporulation once it is completely formed. Is it also thinner when the septum is being synthesized? Because that would tell that the motion of FtsZ is then linked to the synthesis of septal cell wall during constriction itself. So if you look at septal thickness, the sporulating septum is half of the thickness of the vegetative septum as it is being formed. And if you measure the distance which is spanned by the FtsZ bundle during vegetative growth and sporulation, it is also half during sporulation as compared to vegetative growth. So there's a link between the number of FtsZ filaments, presumably, and also the thickness of the septal cell wall. And I think this is a question in the field for really many, many years. We still didn't understand why the sporulating septum might be thinner than vegetative septum and what physiological consequences it might have for engulfment. But now we know that it's probably thinner because you just have FtsZ, which is localized on one side, and it's the number of filaments are less in number, which are dictating the lesser amount of septal cell wall synthesis around the septum. And the thinner sporulating septum might be advantageous during sporulation because, as I told you, later on during sporulation, the mother cell has to just bend and start engulfing the membranes and everything. So it's a very process which requires flexibility, so to say, and that might be relevant for engulfment. And the next thing we wanted to investigate is that what are the factors which might be regulating the divisome architecture? So why do we see a difference in the organization and the thickness of the septal cell wall in vegetative growth and its sporulation? And in order to investigate this, we zeroed in on a protein called SPO2E. So what is special about this protein SPO2E is that it is sporulation specific. So it's only produced during sporulation and not during vegetative growth. And from previous studies, we know that there is some sort of interaction or some sort of a relationship between SPO2E and FtsZ. And what I mean by that is that, so SPO2E has three domains: a transmembrane domain. It's a very large transmembrane domain, a 10-pass transmembrane domain. It has a central FtsZ binding domain, which is also essential for oligomerization of SPO2E. And then it has another phosphatase domain, which becomes important later on when the septum formation is completed. And what we know so far is that SPO2E interacts with FtsZ via its central FtsZ binding domain. And what is special about SPO2E is that super resolution microscopy studies have shown that SPO2E also forms a ring in an FtsZ dependent manner on the 4-spore side of the septum exclusively. So it's not present on the mother cell side at the start of constriction. It's only present on the 4-spore side. And electron micrographs in SPO2E mutant sporangia, so where SPO2E has been knocked out, they also show that the septum is thicker in some of these sporangia as opposed to a wild type sporulating septum. So we wanted to investigate the role of SPO2E, and in order to do that, first we imaged SPO2E knockout strain using cryo-electron tomography. And to our surprise, what we saw was that even though the septum was placed near the pole, if you look closely at the invaginating septum, what we see is that the cell division machinery is present uniformly around the entire leading edge of the invaginating septum. So the pattern of how the cell division machinery is assembled is just like vegetative growth, but the placement of the division is still like sporulation near the poles of the cell. And again, we know that these dots correspond to the cytoskeletal filaments which are encircling the cell. So in pink is the FtsA and in blue is the FtsZ. And this is just a schematic showing how the cytoskeletal machinery is organized in SPO2E mutant sporangia. And then we also measured the thickness of the invaginating septum. And what we see is that these SPO2E mutants are much thicker than sporulating septum, although they're still 10 nanometers or so thinner than the corresponding vegetative septum, which suggests that there might be other factors which are involved in regulating the architecture of the cell division machinery during sporulation. Might be some proteins which are either recruiting SPO2E or are recruited by 2E to the septum. And then we wanted to understand, how SPO2E might be restricting the assembly of the divisome only on the mother cell side. So one of the hypotheses that we had was that maybe during sporulation, initially you have the same number of FtsA and FtsZ filaments as during vegetative growth. But because we know that SPO2E comes in and exclusively localizes on the 4-spore side, so it's possible that it binds to all these cell division proteins on the 4-spore side. And because SPO2E interacts with FtsZ, it might be inhibiting the interaction of FtsZ with the rest of the cell division machinery. And so we, effectively the cell division machinery is not able to carry out its function on the 4-spore side. And then in order to investigate this, what we did was we had an FtsA fluorescently tagged strain, and then we did some time-lapse microscopy on FtsA tagged with neon green. So you see it in green color here. And then the membranes are stained with red using a membrane dye. And what we see is that in these time lapses, you can see first you have a foci which is formed, before the division even starts, on both the sides of the invaginating septum. But as the membrane starts to constrict, what you see is that you see kind of two foci. So one of the foci which stays behind at at the intersection of the lateral cell wall and the septal cell wall, and another foci which sort of constrict. And then we wanted to do some more high resolution imaging. So what we did was we also did some 3D sim experiments. And here you can see much more clearly that the green, which corresponds to FtsA, and again, the membranes are stained in red. You can see that there are two foci. One of the foci constricts with a membrane, which are presumably present on the mother cell side of the invaginating septum. And there are these foci of FtsA which stay behind. Um, at the intersection of the lateral cell wall and the septal cell wall. So leading our credence to our hypothesis is that maybe what is happening is that in wild type sporulating cells, you have some amount of cell division proteins which are left behind and which do not participate in cell division, because SPO2E on that side is interacting, SPO2E on that side is interacting with FtsZ, and it might displace other proteins, although we need more biochemical evidence and in order to support this hypothesis. So just to summarize what we see so far is that we know now we can understand how the cell division machinery, including FtsA and FtsZ filaments, are organized during vegetative growth in Bacillus subtilis, which coats the entire leading edge of the invaginating septum. But during sporulation, these proteins are only present on the mother cell side. And also these are fewer in number, which leads to a thinner sporulating septum as opposed to a thicker vegetative septum. And we understand that SPO2E, which is somehow sequestered only to the forespore side of the invaginating septum, probably constrains the cell division machinery on the forespore side, so that the division happens only from the mother cell side of the invaginating septum. So why are these findings relevant? And I think one of the main things is that now we have a new way to look at cell division machinery, because there have been conflicting views in the field as to what are the major forces or what are the major drivers of cell division in bacteria. And one of the hypotheses which have been existing for a long time is that these FtsZ filaments, they are generating a constrictive force in order to complete cell division in bacterial cells. But if that was the case, then we do not have any of the cell division proteins which are present on the forespore side, but the septum is still constricting from the forespore side. And in recent times, a new hypothesis has come up where it has been suggested that it is only the metabolism of septal cell wall. So the proteins which are making and breaking the different links between the cell wall, they're generating enough energy that can drive the synthesis, or that can drive the synthesis of the septum and continue on with cell division. So I think like the fact that we do not see any cell division proteins on the forespore side which are participating in cytokinesis, and the septum still constricts, gives more credence to the theory that probably it is just the remodeling of the cell wall, the continuous making and breaking of new cell wall materials, which is providing enough energy for cell division to happen. And FtsZ acts like more as a scaffold rather than generating any constrictive force to participate in cell division. And another, I think, a very exciting to place these findings in an exciting concept is that this concept of asymmetry in cell division machinery is a very common theme of development in eukaryotes. So for example, in C. elegans or in Drosophila, like in C. elegans, there are these proteins called PAR proteins, which localized on different sides of the developing embryo in a different manner. So you have some of the PAR proteins which are shown here in blue, and the other proteins which are shown here in red. And because these proteins are localized differently, they position the spindle or the cell division machinery in these cells in an asymmetric manner. And similarly in Drosophila, in which case the cell division machinery is positioned differently, and this leads to generation of cell types which have different fates and different growth later on in the stages. And this is this is what we see here in bacterial cells as well, that they're capable of regulating these complex developmental theme-based ideas by regulating these localization of these protein complexes. And SPO2E here acts as a self-fate determinant because somehow it generates asymmetry by being present exclusively on the 4-spore side, which leads to an asymmetric way in which the cell division machinery is positioned at the septum. To generate cell types with different fates and different sizes before spore in the mother cell. And yeah, with this, I would like to thank all the members of the Pogliano lab and the Villa lab, where I was a PhD student, and all the facilities at UCSD, including the cryo-EM and the microscopy core. And this is an origami of a B subtilis, which I had made at some point, which wasn't related to cell division. But you can see here the DNA being translocated from the mother cell to the 4-spore. And this is really another interesting story. I think these bacterial cells are capable of a lot of dynamic activities which we did not appreciate before. And I think as we evolve techniques to do more genetics and especially higher resolution microscopy methods to visualize the molecular machineries, we can start appreciating the complexity and the diversity of these bacterial cells more and more. Yeah, that's all. Thank you so much.
Thank you for a fascinating talk. That was really interesting to get into the high resolution techniques that you're using. It's really amazing to see like individual proteins within the cell. I'm going to wait a second and let the lag finish to see if there's any questions from the online audience. But in the meantime, I was wondering if maybe you could tell us what do you think needs to happen next on this line of research? Like if you had unlimited funds and a few years and could do anything, where would you go with that?
With the technique cryo-electron tomography in general, or
just follow. Well, yeah, that's interesting too, for sure. But also just following up on what you've discovered here about the asymmetrical division.
I think the next steps are definitely trying to study more the interaction between SPO2E and FtsA, because that is something which we don't understand completely. And also how the interaction of SPO2E with FtsA affects the interaction of FtsA with other components of the divisome. So definitely that is one exciting area of research, which I think will tell us a lot about the mechanisms of more asymmetry. And as I said, although we see that, like the SPO2E, the division septum is very similar to vegetative growth, but the thickness of the septum is still around 10 nanometers or so thinner than the vegetative septum. So it's possible that there are other proteins which are also acting as self-fate determinants during this process of sporulation. And it might be really interesting to explore more of that. So we have some hints from the literature that there might be a protein called div4A, which actually recruits SPO2E to the septum. So there might be involvement of these proteins. And also we know a lot about Min proteins in bacteria, which localize to the cell poles, and then they prevent the assembly of cell division machinery at the poles. So it might be very interesting to study Min proteins in this in these bacteria as well in the context of asymmetrical cell division, particularly because they are also localized near the poles. They are also some way generating asymmetry in polar septation because they are closer to the one side of the pole rather than the other. So I think investigating what more factors might be playing a role in regulating cell division is definitely something which is more exciting. And I think also from evolutionary sides, like we don't know whether this mechanism holds true in other gram-positive bacteria which sporulate and which do not sporulate. And another exciting thing that I was really excited about was that this process of sporulation is restricted to firmicutes, which are these gram-positive bacteria. And the only gram-negative bacteria which is known to sporulate so far is called Acetonema longum. So, and the cell wall structure is completely different in this gram-negative bacteria from gram-positive bacteria. So it might be really interesting to study that even though in gram-negative bacteria sporulates, but its structure of the cell wall is very different. So is it possible that there are mechanisms which are conserved or which diverge in these gram-negative bacteria which sporulate and not? So I think those might be really exciting fields, both trying to decipher more mechanisms of asymmetry, which proteins might be involved, and what are the biochemical and genetic interactions, versus also from an evolutionary perspective to see other proteins or other bacteria which undergo this pathway.
Yeah, that's very, very interesting. Is this a technique that could be used on natural communities? Is this something that you think that like you could take like a mixed ecological community and provided you had
Yeah.
adequate cell density or something like that, and try to get imaging of cell division mechanisms in uncultured organisms?
Yeah, so I think like you can definitely image uncultured bacteria using these techniques or even communities, but then the problem would be like how do you differentiate one bacteria from the other? And the field of tomography, we still haven't come to the point where we have like an equivalent of a GFP tag. So in light microscopy and everything, you can just tag everything with GFP or MJR. You have all these different new tags which are coming up as well to see how proteins are localized and everything. But in bacteria, in tomography, we do not have such tags to visualize individual protein complexes, let alone different bacterial communities. And the other, one of the reasons is that you need something which is electron dense, so that you can visualize it using electron microscopy. And so there have been efforts where you can tag some of the proteins with different iron clusters or something, and iron will just light up in the electron microscope, so you know where you're looking at. So maybe if you want to like for imaging different communities, if it's possible that maybe somehow we have, I don't know a lot about it, but if we can have like a different metal ion associated with different communities, we can light up all these metal ions because they have different spectra in electron microscopy in order to distinguish one thing from the other. But yeah, I think this is definitely like the next step now. Can you go from imaging bacteria in isolation to imaging them in community using high resolution techniques? So yeah.
Great. Well, I don't appear to be any additional questions right now, but that was a wonderful
Mm.
contribution, and really appreciated the presentation quality with all
Thank you
those
so
amazing
much.
videos and reconstructed animations and stuff. Was
Thank you so
wonderful.
much.
Yeah. So thank you for your contribution, and good luck with your new
Yeah.
position.
Thank you so much. It was really nice for hosting me. Thank you.
Our pleasure.
Bye.